Now we are going to do the experiment collecting the DNA using this ripe fruit or banana. So before we proceed to our experiment, we need to gather our materials first. So we will be needing 50 ml tap water, 15 ml dishwashing liquid, 5 ml salt, 100 ml isopropyl alcohol, strainer, 1 ripe fruit or banana, 3 glass containers, and of course, Ziploc. So we're going to mix them together without water, dishwashing liquid, and the salt. And then we're going to stir it carefully until it dissolves. After mixing, we're now going to set aside it for a moment and then prepare for the sample ripe fruit. So we will be using a banana for this experiment. We need to peel the banana first and put it inside the Ziploc bag. So we're now going to put it inside the bag carefully, and then we're going to lock it because we will be going to smash the banana until it turns into tiny pieces. So we will be using our fingers and our hands to smash it into smaller pieces. We'll continue smashing it until it is evenly smashed into tiny pieces. So, the banana in the bag should be completely smashed into tiny pieces until its juices comes out. And then we're going to finish and smash it a little more in order for the other pieces to be smashed up. After smashing for a while, we're going to Pour it in another container, avoiding spilling. Okay, so we're just going to prepare. After placing the banana in one side, we're now going to pour it carefully. We will transfer all the banana juices into another container and then mix it later with the extraction mixture that we did earlier. So I am carefully not leaving any banana in the Ziploc bag so that I can transfer all of the juices into the container. Mixture to the smashed banana. And then we're going to mix it carefully with each other. After being satisfied with the mixture, now we're going to transfer it from another container using a strainer in order to drain the juices of the banana. So now we're going to transfer the mixture using a filter or the strainer, and then we're going to use a spoon to smash the banana against the strainer in order for the juices to drain completely. So we're going to drain all the mixture through the strainer into another glass, forcing all the mixture of the banana or the juices to come out from the strainer. So after draining all the juices of the banana, we're now going to use the 100 ml isopropyl alcohol that was chilled before I started this experiment. So now we're going to pour it into the mixture slowly and carefully. So we're just going to add 50 ml of the isopropyl alcohol to the mixture. And then, after that, we're going to wait for a while until the DNA condense at the top of the mixture. Okay, so here in this, we're now starting to see the DNA or the white clouds forming at the top of the mixture. So we are now seeing that the DNA is now slowly condensating. The white foam or the strings that are slowly gathering at the top of the mixture is what we call the DNA. And then we're going to wait at least a few minutes until we collect it. So after a few minutes, we're now going to collect the DNA that was formed at the top. So it is the condensation of the DNA of the banana. So we're going to get a sample of it. Okay. So the white foamy is the DNA. So we're going to collect all of the DNA that can strangle from our stirrer. Okay, so I'm going to place it in another container in order for us to see how many DNA we've got collected. After transferring all the DNA from another container, here's all what we've got. So there are some DNA that are left from our mixture earlier. So that's all. So thank you for watching our experiment collecting.
Guide question number 1. Do you get more DNA from using your chosen fruit? Why do you think this is so? The chosen fruit in this experiment is a banana. It is said that a banana is a triploid root or plant, and being a triploid plant, it should have a triploid cell wherein it has 3 complete sets of chromosomes rather than the typical pair of chromosomes. Triploid plants are sterile because chromosomes need to occur in pairs to produce viable offspring. It is also said that triploid plants have a lot of DNA inside of their cells, which means that there is a lot for us to extract. With those definitions, I think that the type of plant that we use in the experiment has a lot of DNA. It was then proven by one of our groupmates after doing the procedure correctly.
Question number 2. Do you think you can still get results if you skip any step in the procedure? Why? We think that we could still get results even if we skip any steps in the procedure, but we do not think that we would come up with the reliable results. For us to be able to get a correct and accurate results in anything that we do, for example, this experiment, following the steps and doing it in correct order and way is important. Collecting DNA is a difficult thing to do, thus, skipping steps would practically mean that the results would not be reliable compared to the ones that was done correctly by following the step-by-step procedure.
Guide question number 3. Compare the DNA collected from using your chicks versus the ripe fruit. Chicks and ripe fruit. Plant cells are the ripe fruit, and chick cells are both eukaryotic organisms, thus both can perform special features in the body. For the ripe fruit, plant cells have been differentiated to other cells because of the presences of chloroplasts, cell wall, and vacuole. The procedure of DNA extraction utilizing salt, dishwashing fluid, and ethanol demonstrated to work just on the banana or the ripe fruit, cut, and not the chick cell. Collecting DNA from the chicks are done in swabs. It is said that it was effective but inexpensive way of collecting DNA. It is also considered as non-invasive way of collecting data because it does not use as needles, thus no pain. It is also can be used in newborns. This does not spoil quickly and is good up to 6 months as long as it was stored in a cool, dry storage. It is also considered as the easiest way of extracting or collecting DNA on the lab rather than drawing blood. The nucleic acid from the chick cell demonstrated to be a lot harder to remove. This may be related to the distinction in the test size of the chick and the banana cell.
The experiment was fun and much easier for me because, as I have recalled, we have done it when I was in senior high school and we also used the same process as I did with this experiment. To extract a visible mass of DNA from a fruit tissue, we used a banana because it is the only available fruit in our house. Upon doing the experiment, it was hard for me to drain all the mixture using the strainer because the tissues of the banana accumulated on the strainer's hole. But somehow, I am glad that the result was still a success and we have collected a lot of visible DNA from banana by just following the procedure. I did not expect that we'll be able to take a lot of DNA by just using the mixture of salt, dishwashing liquid, water, and isopropyl alcohol onto the banana. Thus, it was amazing how the isolated DNA from the banana looks exactly how we've studied nucleic acids, in which the DNA resemble a clump of string-like strands that wraps into each other in a helical way. As a result, from doing the experiment, we have gained understanding that the DNA collected differs upon the fruit samples that we will use.